Can we really read and write directly to neural circuits? Imagine being able to listen in on the brain's electrical chatter or even send signals back in. That's the promise of modern neurotechnology. But how close are we to truly reading and writing directly to neural circuits? Right now, scientists use a variety of methods to tap into brain activity. Electrophysiological recordings involve placing tiny electrodes on or inside the brain to measure electrical impulses from neurons. These impulses are the brain's way of communicating. Optical imaging, like calcium imaging, uses special fluorescent dyes that light up when neurons fire, giving a visual map of activity. These tools help us understand how groups of neurons work together. Writing to neural circuits is a different challenge. It involves stimulating neurons to activate or inhibit them. Electrical stimulation is one way, where small currents are sent through electrodes to trigger neuron firing. But a more precise method is optogenetics, which introduces light-sensitive proteins called opsins into specific neurons. When light of certain colors hits these neurons, they can be turned on or off with high accuracy. This technique allows scientists to control neural activity with pinpoint precision. Recent advances combine these reading and writing techniques. For example, optogenetics can be used alongside optical sensors to monitor neuron activity while simultaneously controlling it. This dual approach helps map how neurons connect and communicate. It can even be used to influence brain functions in real time. Applications include brain-computer interfaces that decode what a person intends to do and then send signals to muscles or devices. For example, a person with paralysis might imagine moving their hand, and the system translates that into commands for a robotic arm. Deep brain stimulation systems also use these principles to treat conditions like Parkinson's disease by adjusting stimulation based on real-time brain signals. However, there are hurdles. Many methods requiring invasive procedures, like implanting electrodes deep into the brain. These procedures carry risks and are not suitable for everyone. Researchers are working on miniaturized devices and wearable microscopes to make these techniques safer and more accessible. In data acquisition, capturing neural signals with high resolution and speed is essential. New imaging technologies, such as multi-photon microscopy, combined with artificial intelligence, are helping interpret complex neural activity patterns more accurately. So, can we really read and write directly to neural circuits? The answer is increasingly yes, but mostly in experimental settings. These methods are advancing rapidly and hold great promise for understanding the brain and developing new treatments. While they are not yet widely used outside research labs, ongoing progress suggests that in the future, direct neural communication could become a common part of neuroscience and medicine.